Okay, so that's done. Okay, so let's get started. So this workshop will follow the following structure. So I will start with a short presentation explaining what Ensembl is and what type of data you can find. Then I will run through a demonstration where we will explore Ensembl together. And at the end, you will have time to do some exercises. So the objectives of this workshop are to introduce you to the Ensembl Genome browser, show you the type of data that you can get. Oh, sorry. I'll show you the type of data that you can get in Ensembl, and also how to navigate the website, and most importantly, where to go for help and documentation. So all training materials will be available on our training website, which is training.ensembl.org. And we have our slides in there. We have a link to a living document, some demos, and then also some exercises and answers. So before I get started, I will show you how this training website looks like, so you can get all of the materials there. So I'm going to move to my internet browser, and I will type in training.ensembl.org. All training materials will be under this link here that says training materials at the very top of the page. And our link is the second one, so the one that says Ensembl Browser Workshop, University of Nigeria. So if you click that link, you'll get sent to this page, which is just for today. So we have our slides here on the right-hand side under presentations. We have this living document. I'm going to open that in a new tab and let it load for now. We have a link to a survey. So at the end of the workshop, I'm going to ask you to fill out a survey to see how you found the workshop, and you'll also get a chance to get a certificate of attendance. And then at the bottom of the page, we have some tabs, which are our demos and exercises. So our workshop will be divided into different modules. These are the different modules. And if you click on one of the tabs, you'll see two groups available for you. So the demo, so these are just screenshots of what I'm going to go through on my screen. And then in the second category, we have exercises. So if you open that, we have a number of different exercises. So thank you for filling out the pre-course survey. I could see that we have quite a wide range of interests here. So I tried to put in exercises of different species. So when you do the exercises, you don't have to do all of them. Start off with the ones with the species that you're interested in, and then you can move on to some other species to try out and get to know Ensembl that way. So I'm just going to show you what the living document is. I have this tab open in my browser, and we have this Google Doc with loads of information for the workshop. So we've got a little introduction on the first page. We've got our schedule on the second page. I will make the workshop recordings available on here as well once I've processed them. We've got some golden rules for efficient online training. So I think the best thing to do for today is if you have a question, you can raise your hand, or you can put it in the Zoom chat box. So in the Zoom chat box, or you can write your questions down in this living document. So on page, which page are we on now? Page 4, we have space for questions. So this is really useful when you have any screenshots that you want to share, or if you're getting some kind of error on the browser. So what you do is you just enter your question. You don't have to put your name in. You enter your question, and then I will answer during the times when you're doing your exercises or during the breaks. Okay, so let's go back to the presentation. The training page will be available to you indefinitely. So if you don't have time to do all of the exercises today, you can always do them in your own time later on or tomorrow or in the weekend, whenever you feel comfortable. Okay, so our workshop agenda. The first thing I'm going to do is introduce you to Ensembl. We will look at how you can view regions in the browser. I'll then talk about genes and transcripts. After a lunch break, we will look at variation data in Ensembl and also look at the variant effect predictor, which is a tool that you can use to find the consequences of your variants. Then I'll move on to a comparative genomics module where I'll talk about what kind of comparative genomics data you can find in Ensembl. And then I'll show you this really useful tool called BioMart in Ensembl. So this allows you to extract a lot of Ensembl data at once without the need to know any programming language. Okay, so let's start with a bit of background and an introduction to Ensembl. So first I'll cover why genome browsers are needed, what Ensembl is, and what data we offer. And then I'll briefly go through how genome assemblies are made. And then finally, I'll give you a brief demonstration where you can either follow along on my screen or you can do it on your own screen along with me. And then at the end, you'll have time to do some exercises. Okay, so in 1977, Fred Sanger and his team sequenced the very first genome, which was a bacteriophage. And this really kickstarted the era of sequencing and also sequencing much more complex genomes. The very first draft of the human genome was published in 2004. This was a project that was started back in 1990. And since then, many, many more organisms have been sequenced. And today we can sequence a complete human genome in a matter of days. So with this, Ensembl was launched in 1999 with the goal to automatically annotate this first draft of the human genome and also integrate it with other data to make it publicly available. But why do we need genome browsers like Ensembl? So with sequencing becoming cheaper and faster, genomic data is piling up very, very quickly. But when sequencing data is produced, you get something like this, which is just a bunch of letters. So trying to read a genome without annotation data would be like trying to read a new language to you that you don't understand. So it wouldn't mean anything to you. And this is what genome browsers are there for. You can use genome browsers like Ensembl to map annotations to genomic data. And Ensembl brings together information from a wide range of other databases in a single site. So other genome browsers that you might know are UCSC and NCBI. And I also want to emphasize that at Ensembl, we do not do our own sequencing. So we simply take publicly available sequencing data and we use our own pipelines to annotate the genomic data. So the Ensembl browser is dedicated to vertebrate species. But as we know, vertebrate species are only a fraction of the different organisms in the world. So you can find some non-vertebrate species in Ensembl, for example, fly, worm, and yeast genomes. And this is only because they're in the Ensembl main site only because they're model organisms. But in general, for any non-vertebrate species, you will have to visit our sister site, which is called Ensembl Genomes. So this is divided into different taxa. So we've got Ensembl plants, Ensembl invertebrate metazoa, Ensembl bacteria, protists, fungi, and we also have a COVID-19 site. And each of these taxa have a different color coding. But the site themselves, they are structured very similar to the Ensembl main site. So once you're familiar with the main Ensembl browser, you'll have no problem navigating any of the other Ensembl Genomes sites. So what can you find in Ensembl? You can get reference genomes, which are annotated with different features. So we annotate a number of different things in Ensembl. These include genes, variants, promoters, binding sites, and loads more. One term you're gonna come across to quite often is the word gene build. And a gene build is just, it means genes mapped to a genome. So we have gene builds available for all of the species in Ensembl. We also have variation data in a number of different species. We have comparative genomics data, such as alignments, gene trees, homologs. There are also regulatory builds available for human and mouse. And you can also use a number of different tools in Ensembl to export. So for example, BioMart to export a large amount of data, or VEP, the variant effect predictor, to determine the consequences of your variants. So you can access Ensembl via the web browser, which we will do today, but you can also integrate it into your own pipelines via APIs. And most importantly, Ensembl is completely open source. So you can use it freely in your own research. So we have about 300 vertebrate species available in the Ensembl main site, and in Ensembl Genomes, so the sister site that is dedicated for non-vertebrates, we have over 2,000 eukaryotic and over 3,000 prokaryotic genomes available. So this table just shows you a little bit of a summary of the different Ensembl sites that we've got. So we've got the Ensembl main site. This was released back in 2000. We've got vertebrate species available in there, and the annotation is done completely by us, so Ensembl. We've got the Ensembl Genomes site, so the one for non-vertebrates. This was published back in 2009. So this is dedicated for any non-vertebrate species. And the main difference between the main site and the Ensembl Genome site is the annotation method. So for the Ensembl Genome site, we actually do the annotations in collaboration with scientific communities. Whereas the Ensembl main site, this is done by us. And then we also have the Ensembl COVID-19 site, which you might have guessed was published back in 2020. Okay, so because genomic data is constantly evolving and changing, Ensembl is updated every 3 months to represent the most recent findings. Behind every update, a lot of work is involved. So this includes the addition of any new genome assemblies, updating underlying software, updating gene sets, updating comparative genomics analyses, and so on. The current release is 109. So this was published back in February 2023. And this is for the Ensembl main site. And Ensembl 110 is expected to be released around the end of May. So this is for the Ensembl main site. For the Ensembl Genome site, because it is a little younger than the main site, the release number is different. So for the Ensembl Genome site, the current release is 56, published back in February 23. And the next release, 57, is also expected end of May. So because the Ensembl sites are not static, you always want to make sure that you write down which release number you're working with. And I will show you how you can find out which release you're working with during the demonstration. And then, oh yes, we also keep older versions of Ensembl. So for example, if you need to work with an older release, let's say Ensembl Genomes 55, we keep the archives available for up to 5 years. So if you need an older release, I will show you where to find these in the demonstration. You can get them in the Ensembl browser as well. Okay, so new genome assemblies are published all the time. And because of that, apart from Ensembl and Ensembl Genomes, we also have the Ensembl Rapid Release browser. So this is updated every 2 weeks, and the browser includes genomes with gene annotations, and the only other tool you can use is BLAST. We don't have BioMart on the rapid release site, and there's also no variation data available. And this is because the website is updated much more frequently. So there isn't much time to to add these genomes to BioMart and add variation data to it. Oh, sorry. Oh, sorry, my computer is playing up a little bit. Sorry about this. So I can just see that someone asked for the schedule. I've actually forgotten to add the training page link to the Zoom, so that's a good time to do it. So you can find all training materials in this Zoom link, and there is a link to the living document, which also includes the schedule for today. Okay. So yeah, rapid release site updated every 2 weeks. We only have gene annotation available. The only other tool is BLAST, and we don't have BioMart and variation data because it's updated much more frequently. So let's look at genome assemblies. So the human reference genome is not a genome of a single person, and it's not a consensus either. It's actually made up of fragments from different individuals. And each fragment represents a real person's genome, making the reference genome sort of like a jigsaw puzzle or a hybrid genome of different individuals' DNA. It's not a consensus genome. So a consensus genome would be made up of the most common sequences found in the pool of sampled sequences. And that's not what it is. So just remember that the reference genome is a hybrid genome. And talking about sequences, so let's just recap how genome assemblies are generated. So here you see a simple diagram of sequencing reads. So this is one read, second read, and so on. So sequencing data is assembled by matching up any overlapping reads. So we put the reads together and we see which regions match up. So for example, this first read matches up with this part of this read, and then the next one matches up with this. And that's how genome, the reads are assembled. So what this makes up is a contiguous sequence. This is also called a contig. And a contig is just a consensus region of DNA. These contigs are then assembled into scaffolds. So a scaffold is a run of contigs or an assembly of contigs without any gaps. And then finally, if there is enough mapping information available, scaffolds are assembled into chromosomes. So genome assemblies are not a perfect representation of the genome. And it's just important to remember that there are still some errors and gaps in genome assemblies. Okay, so what can you use Ensembl for? To summarize, you can explore genomic regions in the browser, you can learn more about genes, you can learn about variants of interest, you can find out about any genetic background of traits. All right, moving on to the first demonstration. For this, I will show you how you can navigate the Ensembl and also the Ensembl Genomes homepages and find out more information about any species you're interested in, and also more details about the genome assemblies across the different sites. So please feel free to follow along on my screen or follow along on your own screen. So I'm going to move to my browser. Excuse me. So I'm going to move to my browser and I will open a new tab. And what I will show you first is the Ensembl main site. So the site that is dedicated to vertebrate species. And to get there, all you need to type into your address bar is ensembl.org. That will open the Ensembl homepage. So here at the very top, we have our navigation bar. So this is static across all the pages, any pages that you go to. They just have some shortcuts to some popular tools. Also a shortcut for download. So if you want to download whole genome data, for example, the FASTA file of a species, you can get that from here. There is also a link for help and documentation. So if you're interested in what type of data you're looking at the moment, or if you're not sure how the data was generated, you can find that here. And we also have a link to our blog. So our blog would have posts like updates on any Ensembl data. And we also release job openings on there as well. If you use Ensembl quite regularly, I suggest you register for an account. So this is free. You can delete your account at any time you want. But this just makes it easier if you're, for example, looking at a particular species quite often. So if you focus on the bottom of the page, there is the section that's called Favorite Genomes. There's a section that's called Favorite Genomes, and you can change the favorite genomes in here according to whichever species you're interested in. I'm really sorry, I'm getting some feedback here. Could you mute yourself?
I doubt if she's aware of this. Maybe you can mute her as well, mute everyone. Yeah.
Okay, I think she's done that. Yeah, because it's she's muted now, yes. Okay, cool. So we have, if you use Ensembl quite often, you can register. And the things you can do when you register is, for example, change the section of favorite genomes. So if one of your genomes of interest is not human, mouse, or zebrafish, you can change this to, let's say, panda if you're studying panda. Okay. So then here in the right-hand corner, we have this search bar. So this is a site-wide search bar. If you're looking for documentation, you can use this to search for it. Then going down, we have some information about Ensembl, so just a little summary of what Ensembl is. And then we have the section that gives you the updates on this current release. So we are currently on Ensembl release 109, published back in February 23. And these are just some highlights of what we've changed in this release. Scrolling down, we have a link to the rapid release site. So if you're ever looking for a species that you cannot find on Ensembl or Ensembl Genomes, it might be available in Ensembl Rapid Release. Next, scrolling down to the very bottom here on the right-hand corner, we have a link that says View an Archive Site. So if you click that link, you get this little pop-up window, and here is where you can find all of our available archives. So let's say you worked with an older release like Ensembl 104, you can just click that archive site link at the very bottom right corner and get to the archive site using these links here. I'm going to close this pop-up window for now. Moving to the left-hand side, here in the left-hand corner, we have again our release numbers. So Ensembl Release 109, published in 2023. And then scrolling up to the top of the page, we have our species box here. So here in this box, you can find a link that says View Full List of Species, and this is where you can search for your species of interest. So we've got common names here in the first column, then the scientific name, any IDs, so assembly ID, accession IDs, and then these three columns at the very end are, I think, what are really important. So these show you what type of information we have for each of the species. So it's going to tell you if we have gene annotation available, so gene builds. It will tell you if we have variation data available or regulation data. You can sort these this table according to the columns. So if you just click on the column names, it will sort the table accordingly. So if you're looking for variation, if you want to find out what type of variation data we have available, you can sort your column, and then you can see everything that has a Y means we have, yes, we have variation data available. You can download this table and view it in your local machine or on Excel if you wish, using this link here at the very top right corner. You can either download the whole table, or if you applied any filters, you can download that table. And if you're searching for a particular species, you can use this search bar. So let's say you're looking for human. You just type in either the common name or the scientific name, and then you can see that the table gets filtered down to whatever you typed in there. Okay, so if you want to learn more about species, you can click on the common name. And here, where you are now is called the species information page. So on this page, you'll find all the type of data we have for human, in this case for human. So we've got genome assembly data available. So these just gives you some information on where we retrieved the assembly from, and also any summary statistics. We've got here, you can see we have gene annotation data available, we have comparative genomics data available, variation, and then also regulation data. For each of the different data types, you can always view some examples. So here in these little icons, if you click on them, you can view some example regions or features. Under genome assemblies, there is a link that says more information and statistics. So if you click that link, you can find out a description of the assembly, but also some summary statistics here on the right-hand side. So for example, this assembly is the GRCH38, and this assembly was taken from the INSD database. So this is the assembly ID that you can find here. And then we also have some numbers like number of coding genes, number of pseudogenes, and so on. If you're working with one of the older human genome assemblies, so let's say GRCH37, we also have a dedicated site for that. So to get there, you can just use this drop-down menu here on the left-hand side, select GRCH37, and then click Go. And that will send you to the Ensembl GRCH37 dedicated page. So this is the older human genome assembly here. Okay, I'm going to go back to my to the Ensembl main site. And I will show you the Ensembl Genome site now, so the one that is dedicated for non-vertebrates. So to get there, you can either use these shortcuts at the very bottom of the page here on the right-hand side, or you can type into your browser ensemblgenomes.org. Okay. And this is our dedicated Ensembl Genomes homepage. So if you're interested in plants, you just go to plants. We've got invertebrate metazoa, protists, fungi, and bacteria. If you're not sure which taxon your species is in, you can also search across all of the sites. So let's say you're interested in a species called Xylella. You can type that in and then click Go. And that will show you all the different entries for that species across all of the sites. Okay, going back. So these pages, these non-vertebrates pages are all structured quite similarly to the main site, except the bacteria one. So I want to show you how you can look for a species in bacteria. So in bacteria, we have 10,000s of species. So we don't have that sort of drop-down menu that we would have on the main site. Instead, what you would need to do is search for your genome using the search bar at the top of the page. So if you're interested in E. coli, for example, you type in the name, and you can see that that you have this drop-down menu open now, and you can find all the different E. coli genomes that we have available this way. So if you have a particular strain you're interested in, I think K12, is that a strain? Yeah, SK12. Sorry, no, it is K12. So you can just type in your strain name and then look for it from the drop-down menu. Alternatively, you can view the full list of Ensembl bacteria species. So there is the link here underneath this gray box. If you click that link, it will send you to this massive table. So here you can also search for your genome of interest. So let's look for E. coli again. And then it takes a bit of time to update, but then you can see the table now only has E. coli filtered down. And you can also see how many entries we've got for E. coli only. So we've got 1,259 for E. coli alone. Okay, so that's Ensembl. So I think I've shown you everything I wanted to show you for now. So we've gone through the through the homepage. We've looked at how you can find information about a particular species. What we're going to do now is do some exercises. So if you go to the training page, I've got the link. I've got the tab still open on my browser here. At the very bottom of the page, there is a tab that's called Species and Genome Assemblies. And under exercises, I think you have 5 different exercises. So one for bacteria, one for fungus, one for plants, another plants, and then one for the main site. So start off with the exercise that sounds most interesting to you, and then move on to the other exercises. I'm going to give you, let's say, 15 minutes for this, just to get you familiar with Ensembl and also just browse around a bit to have a feel of the website. So we're going to do the exercises now, and we will come back at, let's say, 10 past. Okay, it's 10 past now. Would you like some more time to continue the exercises, or will I move on? Okay, I think I will move on in this case. So the exercises will be available to you indefinitely. So if you do one more time, please feel free to do that during the breaks or after the workshop. But for now, I will move on to the next module, where I will talk about how you can view regions in detail on the browser. Does anyone have any questions before I start? Okay, so if you do have questions, please put them down in the Zoom box or in the living document.
I have a question.
Ah, yes. Hi.
Hello.
Hi, yes, I can hear you.
Okay.
Okay, I think I'm getting a bit of feedback from you. Would you mind putting your question in the chat box?
Hello.
Hi.
Hi.
Yeah, I'm getting a bit of feedback here. So sorry. Okay, I've just muted you because the feedback was getting quite bad. So if you could put the question in the chat box, or if you're in the classroom, you can just use the classroom. Ah, okay, you've just typed it in there. So you said, please, can you give us more time so we can interact and perhaps be all at the same speed? Of course, yeah, I'll give you another 10 minutes just so you have more time to explore the browser. So let's come back at 10:25. And then we'll see where we're at.
Hi, Luis. We can continue now. We are ready.
Okay, perfect. Thank you. All right. Okay, are there any questions before I move on? Okay, no questions so far. If you have questions, please. please put them down in the Zoom chat box, or in the living document. So for this next module, I will show you how you can view a specific region of a genome in the Ensembl browser. So for this, I'm going to go straight to a demonstration. And we are going to look at a region in E. coli, and the region is these coordinates. So first of all, I will copy these coordinates and put them in the chat box so you don't have to type it in. And next, I will move to my browser. So I'm going to move to my browser and open my Ensembl browser tab. I've already got this open here. So I will go to the home page first, so we all start at the same spot. And the first thing I will do is search for my my species of interest. So I've entered the species, the E. coli strain in the chat box. And what you can do is you can just copy that and type it into this, paste it into this search box here in the gray block. So I've done that. And now it's loading, and you can see that from our drop-down menu, we only have this available. So we'll click this species, this strain. And you are now on the species information page of the K12 E. coli strain. So to search for a feature or coordinates in this strain, we want to enter our coordinates into this search box here under the name. So I'm going to copy the coordinates and paste it in here. And these coordinates have a very particular format. So it starts off with chromosome. So this would be the chromosome number if a genome would have multiple chromosomes, but in E. coli it's only one. So it says chromosome, followed by a colon, followed by the start position, followed by a minus, followed by the end position. Once you're happy with the coordinates, you can click go. And now we're just waiting for it to load. So if you're searching for coordinates in a particular species, by default, you land on the location tab. So we can see we're on the location tab by looking at the top left-hand corner here. So under the species name, we have our location tab. And in this location tab, we have different displays or views that we can choose from. We land by default on the region and detail view, but you can also look at the chromosome summary or the whole genome. But for now, we will look at the region and detail view. So this view is divided up into 3 different resolutions. We've got the chromosome view. We've got an overview of our region. So in this case, it's a 50 KB overview. Our region of interest is this red box that that's highlighted here that I'm showing on my screen. And then further down is what we call the region in detail view. So this is just the region that you've searched. So let's start at the very top at the chromosome view. So here, our region of interest is highlighted in this little red triangle. If you hover over it, you might be able to see it a little better. You can, so these views are interactive, so you can use your mouse to click around. So for example, you can actually mark a region by clicking on these little circles on the handles, and moving the handles highlights different regions. You can also just use your mouse to click on any region on the chromosome and move the handles that way. Next, we have our 50 KB view. So this overview depends on the on the size of the genome. In our case, it's 50 kb. For example, in human, I think this would be 10 MB. I'm not sure, but depending on the size of the genome, this view might be a different scale. So in this view, we have our one track, which is called the genes track, and track is a very specific genome browser term. And basically what it means is features mapped to the genome. So in our genes track, we have genes mapped to our genome. So our K12 E. coli genome. Each of these blocks represent a different feature. So this, for example, is the GLCD gene. This one is the YGH5 gene, and so on. You can also see less than and sometimes also a greater than sign next to a name. And this just represents the direction in which the gene goes. So if it's a greater than sign, like this GPR gene in this track, it means it's located on the forward strand. And if it has a less than sign, like this EXPD gene, it means it's located on the reverse strand. Okay, so this view is also interactive. So if you want, you can change your mouse actions here on the top right-hand corner. You can select a region, or you can drag or scroll across the region. So at the moment I've got this select a region action. So what you can do is you can highlight a region. It gives you a little pop-up, and there are two options you can do. So you can jump to the region. What this will do is it will update our very bottom view, or you can just center here. So that will update this view here. I'm not going to do anything because I want to show you the region that we've just searched for. So we'll close this pop-up. Okay, scrolling down, we have the region, the actual region that we've just searched for. You can double check that by looking at this location search bar at the top of the view. But here is where we see more details about our region of interest. So the first thing you will notice is you have two genes tracks. So we have one here and one here. So the reason for these two tracks, it just indicates one track showing genes on the forward strand and the other track showing genes on the reverse strand. I usually use the context track as sort of a centerpiece. So I always focus on the context track. So this is just the genome assembly track. And any features located above the context track are located on the forward strand, and everything located below the context track are located on the reverse strand. You'll also notice that there are different colors. So we have red, we also have this gray here, and at the bottom of the view, you will see a legend for the tracks. So for our genes, red represents protein coding genes, and this gray represents pseudogenes, so a non-protein coding gene. Here as well, you can click on the different features. So if you're interested in this particular gene, for example, you can click on that block and you can find information like the gene name, the protein product, the location of the gene, so forward strand, the size, and then the exact coordinates as well. Let's close this little menu. What you can do in this view is you can add more tracks to the view. So if you're interested in very particular data, you can add more information to this data. So there is a button in the top left-hand corner that says Add Remove Tracks. And we're going to click that. And what we have is this, this menu that pops up. And here on the left-hand side, we have different categories that we could choose from. So we've got sequence and assembly information, we've got genes and transcripts information, mRNA protein alignments, repeat regions, any other information, and then we have some other options as well. Here on the on the right-hand side, you will see all the active tracks that are turned on at the moment. So we have the context track, sequence, genes, and so on, all the scales and rulers and all of that. If you're not sure what these mean, you can always go to the right-hand side and click on this icon, so the information icon. And it just gives you a little description of what the track is. You can search for a track, so if you're not sure which category it's in, you can just use this search bar in the top left-hand corner. But for now, let's just explore some of the tracks here. So let's say you would like to look at, you would like to map all of the start and stop codons in the genome or in the region. This is under sequence and assembly. And this fourth track, it says start or stop codons. So I'm going to click that. And when you click this square, it means that the track is turned on. Let's also turn on another track. So just to see, so you can see how it looks like. Let's open the repeat regions. So I want to see all of my repeat regions in the browser. So I'll just click all repeats, and that's now turned on. To save and close the menu, you just click on this check icon in the top right-hand corner. Okay, and then you can see that our view is just loading now, so we're just waiting for Ensembl to load that. And now we have our new tracks available in our region. So we have these start and stop codons here. Again, 2 tracks because of features located on the forward and reverse strand. And then here at the very bottom, we have our second track that we turned on, so the all repeats track. So this shows, if you're not sure, you can click on it. It shows all of the repeat regions in our region of interest. Similar to the genes, each of these blocks represent a different feature. You can zoom into a region. So let's look at this particular region. To mark, you can just click and drag your mouse and then click jump to region to open. That will update our entire view, and it will just zoom into that region more, so you can look at it in more detail. So you can see we can only see this gene that we've just highlighted there. To go back to your previous view, you can just use your back button in your browser. Okay, and you'll see our view has just reset. You can move around the tracks. So if you wanted to compare certain regions together, features together, you can move them up and down using your mouse. And then, of course, you can delete a track as well by clicking on this X icon after clicking on the track name. And that deletes the track here. Now, if you wanted to share your configurations with your colleagues, what you want to do is you want to generate a URL. So if you were to copy the URL from your browser address bar, they might not see the exact same configurations that you've just selected. So instead, what you want to do is you want to scroll up. And here on the left-hand side, we have this button that says share this page. And that generates a URL for you that you can copy and then share with your colleagues. And then, of course, you can export the image as well. So you can export this overview by clicking on Export Image, or you can export the region and detail view by clicking, again, Export Image. Okay, so that's all I've wanted to show you for this region and detail module. So now we have time for the next set of exercises. So we're gonna, you're going to move on to the exploring genomic regions exercises, where you will have multiple different species available again. So you can start with the one you're most interested in and then move on with the others, just to get a feel of how you can navigate the region in detail view. So I'm going to give you 20 minutes for this. So let's come back at 11 and then we'll see where we're at, and then we will take a little coffee break. All right, it's 11:16 now. Is everyone back? Can I move on, or would you like another 5 minutes? Okay, I think we will move on in that case. So we'll do one more module, and then after that, after we finish the exercises, we will take a lunch break. So let's move on. Okay. So for this module, I'll be talking about genes and transcripts in Ensembl. I'll explain the gene view in more detail and how genes are annotated in Ensembl. I'll also talk about how stable IDs are how do you call it, structured, and then we will also talk about gene ontology, which is what describes the gene function. And then we'll move on to a demonstration, and you'll have time to do some exercises. Okay, so let's talk about gene annotation in Ensembl. Genomes that are annotated with genes are referred to as gene builds, which I mentioned earlier. And this is a term you'll come across quite often when working with genome browsers. There are two independent annotation methods that we use in the Ensembl main site, so the one dedicated for vertebrate species. Both rely on valid biological experimental data. In Ensembl, gene annotation is only mapped when there is experimental evidence that a gene is transcribed. And we have the gene automatic annotation pipeline, which is applied to all vertebrate species. And we also have the Havana manual annotation pipeline, which is done by a group of annotators, so people, and this is applied to human, mouse, partially zebrafish, and rat. But unlike the Ensembl main site, we do not actually carry out primary annotation on the Ensembl Genomes sites. So in the Ensembl Genomes sites, which are dedicated for non-vertebrates, we actually import gene models from publicly available archives. And Ensembl Genomes is involved in collaborations from which annotations are imported. So they might use very similar methods like us, so maybe some automatic annotation or manual annotation, but they could also be annotating genes in those genomes using homology or ab initio predictions. So we import data for each assembly in the Ensembl Genome site from different databases. For example, for Fusarium, we take genome annotation information from Rothamsted Research. So this is an institute in the UK. For wheat, Triticum aestivum, we take information from the INSDC RefSeq collaboration, and the genes annotated here are done by automatic annotation and also homology predictions. So it really depends on the species in the Ensembl Genome site where we take that information from. So we either import annotations from specific consortia, or we perform annotations in collaboration with scientific communities. We import genome assemblies from a number of different databases. So these can be any database under the INSDC or International Nucleotide Sequence Database Collaboration. And INSDC includes the European Nucleotide Archive, ENA, NCBI, and DDBJ, which is the DNA Data Bank of Japan. So in this slide, what you see is the region in detail view, which we explored during the demonstration earlier. So let's just look at this in a little more detail. If you search for a specific gene, the gene will actually be highlighted in this green color. So like here on the slide, we already know that the view is made up of different tracks. So we have the genes track, one above the contigs and one below the contigs track. And each track represents a different type of feature that is mapped to the genome. Each feature is shown as some sort of block, colored block. So this is one feature, this is another feature, and so on. And the features can also have different colors, and that just represents different types of features. So in this genes track, let's focus on this genes track below the contigs. So we have a red and gray, and I already mentioned earlier that red represents protein coding genes, and the gray represents pseudogenes, which are non-protein coding. You can also find a description of the different tracks. So if you're not sure what the track is, what information the track is showing you, you can click on the track and you can find information that way. And in this sort of pop-up menu, what you also get are different options. So you can change the style, how you want to see it in the browser. You can also delete the track this way. You can click on the individual features. So we did that as well. We clicked on a gene, and that's how you can find more information about the gene. So we have the transcript ID, the gene ID, protein ID. We've got the type of gene. So in this case, this is protein coding. We also have the orientation of the gene. So in this case, it's on the reverse strand, and the coordinates and the size of the gene as well, and the protein coded from it. And then most importantly, we also show you how the gene was annotated. So at the very bottom, we have this section that's called annotation method. And here, if you click on each of the genes, you can find out how the gene was annotated. So in this case, protein coding genes were annotated in ENA, which is the European Nucleotide Archive. Okay, so I want to mention gene ontology, or Go terms. So just like in any language, there can be multiple terms for the same biological function of a gene. So for example, one person might talk about innate immunity, while another person might talk about non-specific immunity. But we both know that we're talking about the same thing, so innate immunity. But to tackle this, to make sure that we're actually indeed talking about the same thing, the scientific community have come up with Go terms, gene ontology terms, which are just a standardized way of describing a gene function, to make sure that we are talking about the same function. So Go terms are made up of unique Go IDs, which you can see here in blue, which make them more easily searchable. And then it also has a human readable description. So in this case, we have this Go ID for metabolic process. And Go terms are structured in a hierarchy. So a Go term can have a parent term, which describes the wider function of the feature, but it can also have multiple daughter terms, which describe more detailed functions. There are three categories or we call them aspects of Go terms. So the biological process describes what the gene does. The molecular function describes how the gene does it. And the cellular component describes where the gene or protein, the product, is located. So let's take human cytochrome C, for example. The biological process of human cytochrome C is oxidative phosphorylation. So this is what the gene does, or the product, the protein. Then the molecular function would be oxidoreductase activity. So it does it by oxidoreductase. And then the cellular component, or the location, would be the mitochondrial intermembrane space. If you are interested in the more technical side of annotation in Ensembl, there is a paper out that describes this in more detail as well. Okay, so we're going to look at the LAC-Z gene in E. coli K12 again. And what we're going to do this time is we're going to find out information about the gene itself and also its product. So for this, I will move to my browser and go to my Ensembl tab. And then what you can do is you can click the E. coli name at the top of the page. So let's click that. So someone's just asked a question in the chat box, and they've asked, does Ensembl also do metabolomics analysis? Yes, but only for certain species. So for Ensembl Genomes species, this is very limited. I need to find out which species exactly, but we do them for the Ensembl main site, so the vertebrate species. And if you want, you can tell me which species you're interested in, and I can find information whether we have data for that or not. Okay, so I've just clicked on the E. coli name. We're still with the same strain, so K12, and I've clicked on the name so I can do a species-specific search. The gene we're interested in is LAC-Z. So I will enter this in the search bar here. So if you enter LAC-Z and then click Go, that will search for the gene in E. coli K12. Okay, and then you will get a search results page. Sometimes you would have multiple results for a gene because gene names can be there can be multiple names for a gene, multiple names for a gene. So sometimes you'd get multiple results. But we always give you the description so you can make sure that that's the gene you're actually looking for. Sorry, my mouse is playing up today. Hang on. Okay, right. So we have the description. It's a LAC-Z is a beta galactosidase. We have the gene ID. We've got the species, so just making sure it's E. coli K12. And then we have the location, and then any gene trees if there are any available. We'll talk about comparative genomics data in the afternoon today. You can click on the location link directly, so that will send you directly to that location tab that we looked at earlier. But what we want to do is find out information about the gene. So we'll click on the gene ID link. And if you focus at the top of the page, you can see that we now have a new tab that is called Gene LAC-Z. So before we were on location, and now we are on Gene LAC-Z. Here on the left-hand side, we have different displays available for our gene. So we can find information about the sequence, ontologies, so the function, comparative genomics data, and then also any external references. Underneath, we have different options, configurations that you can choose from. So you can configure this page similarly to the region and detail view from earlier. You can add tracks. You can export data. You can share the page. So if you want to share this gene with your collaborators, you can generate a URL here. And then if you have an account with Ensembl, you can bookmark the page so you can find it easily later on. Here on the right-hand side, we have some summary information about the gene. So a description, the exact coordinates, the orientation. So it's located on the reverse. And then also how many protein families this gene belongs to. So in this case, 2 protein families. There is a transcript table here. This is less relevant for prokaryotes, but for eukaryotes, we would have multiple transcripts. We could have multiple transcripts for each gene. So we only have one for this because it's bacteria. Okay, next we have some links to databases. So if you would like to look at the LAC-Z gene entry in UniProt, you can find a link directly there. And then also, again, the annotation method. So here, this gene was annotated in the European Nucleotide Archive, or ENA. And you can find a link to that direct entry by clicking ENA. Okay, so if you wanted to look at the sequence of the gene, you can click on sequence here on the left-hand side. And what we're getting here on the right is our LAC-Z sequence in FASTA format. So we know it's in FASTA format because it starts off with a greater than sign, followed by a description or name of the sequence, followed by the actual sequence itself. So it's quite a small gene because you could scroll down and see the entire sequence. There are different highlights available. So for exons, again, not so relevant for prokaryotes, but for eukaryotes, it would be. We highlight the exons. So the red would be the exons within our gene of interest, so LAC-Z. And then the peach colored would be exons of neighboring genes, so flanking genes. By default, we show you 600 base pairs up and downstream of the gene, but you can change that. So if you only wanted to see the gene itself, you can click on configure this page on the left-hand side. And this opens a menu, and here you can change some configurations. So let's just change this to, I don't know, 50 and 50 again. So we only see 50 base pairs up and downstream. And then you can also choose to show any line numbering. So it's easier for you to look at the sequences. So you can choose relative to the sequence or relative to the coordinate system. I'm going to select coordinate system. So this is relative to the entire E. coli K12 genome. And once you're happy, you can click this check icon in the top right-hand corner to save and close the menu. And now you'll notice that the flanking genes have indeed decreased. So now we're only seeing 50 base pairs up and downstream of the LAC-Z gene. You can download the sequence. So there is a button at the very top that says download sequence. This opens a pop-up window again. And here you can select different file formats. So you can select between the FASTA format, which we're seeing in the browser, but you can also select the rich text format, or RTF. And this is how we're seeing it on the browser. So the FASTA format would have no colors. It would just be text-based, whereas the RTF would have the nice colorings. So it'd be easier for you to look at the sequence. But most of the time, you're only going to need the FASTA format because that's the input for a lot of different bioinformatics tools. If you're not sure what the formats look like, you can scroll to the bottom of the menu, and you have these guides or previews of the different formats. So again, FASTA format looks like this, and then RTF would look like this. To download, you can click download, or if it's a very big file, I suggest you compress it first and then download it because it's much easier. So you can do that and download it save it directly on your local machine. I'm not going to do that, so I will close this menu for now. Okay, so let's look at the different functions of the gene. So here on the left-hand side, again, under ontologies, we have these three aspects of Go terms, biological process, so what the gene product does, molecular function, so how the gene product does it, and then cellular component, where the product does its function. Let's look at the biological process, so what the gene does. Okay, and now here on the right-hand side, you'll find a list of all the different Go terms associated with our LAC-Z gene. In this case, we only have two Go terms associated with it. So it's the first function is carbohydrate metabolic process, and the second one is lactose catabolic process. There's some more information about the evidence. So if you hover over the evidence, it gives you a description of what it is, and then you can also view it on the chromosome in the last column. If you want to find more information about the protein, about the product, so the protein, we want to open the transcript table and click the transcript ID in the table. So we already had the tab open. If you look at the very top, we have this transcript LAC-Z1 tab. And on the left-hand side, our views or displays have changed a little bit. So now you can find protein information about our gene. You can look at the protein summary. So if you click protein summary here on the left, we have information about the different domains of the protein. So each of these lines represent a different type of domain. You can click on them to find more information about them as well. And then also the position and how calculate how long it is. If you want to download the sequence, there is a link for that as well. So here on the left-hand side, you can click protein sequence, which will give you the protein sequence of the LAC-Z gene. Similar to the sequence before, we can download this as well, but you can also perform a BLAST. So this is an alignment tool that you can do. And what it does is it aligns this sequence, so our sequence, to our query sequence to a database of whatever species you're interested in. So you can find, what you can do with it is you can find if this sequence exists in any other species, and if it does, how similar are the sequences. Okay, so if you want to look at any entries in external databases for our protein, LAC-Z protein, you can click on General Identifiers, and you will have direct links, direct entries to the database here. So if you're looking for a particular one, you can search for it in the first column, and then click on the link on the left-hand side. Okay, so there's been a question. So the question is, are the exon letters for proteins? So if I just open the protein sequence here. So each of these letters are the different amino acids. So they are protein sequences. I hope it makes sense. Oh yeah, one thing I forgot to mention was if we go back to the gene tab, often what you will find for each of the types of data that we show is this question mark next to a name. So if you click on the question mark, it opens a new tab and it will give you a summary of what the data is that we are showing you. So I've just clicked the gene summary question mark. So this is the help page for gene summary. You can find information about the different types of data we're showing you. So the name, the biotype, and so on. Okay, so this is how you can learn more about genes and proteins in Ensembl bacteria. So now we have time for our exercises again. I'm gonna move to the training page, and the category we're looking at now is genes and transcripts. And again, we have loads of exercises here. So start off with the one that you're interested in. And I'm gonna give you, let's say 20, 25 minutes to do this, and then we'll come back, see how it goes. And if you'd like more time, we'll give it more time. And if not, we will take a lunch break. So we'll be back at 12:05 then. Okay, so there's been a question in the chat box, and I think it's easiest if I show it to you. So the question is, how do we get the sequence of the different protein domains of the LAC-Z gene? So let's go back to my browser and to my Ensembl tab. And I will go to the transcript tab to look at the domains. So this was under protein summary here on the left-hand side. And what you can do is, if you want the entire, all of the domains, you can click on export data here on the left. And that will allow you to download the annotation file of the gene. So you can get the annotation file in GTF or GFF format. So depending on your downstream analysis, you might want a very specific format. And that will also highlight all the different domains and where they start. So select your format and then click next, and then that will download it directly. Well, you select your sorry, you need to select where you want to view it first. So you'll view it on the browser as a text, or do you want to download it as a compressed file? And then you click on one of them and that will download it directly onto your local machine. Another thing you can do is you can click on a particular domain you're interested in and open the entry to that external database from it. So that entry was taken from Interpro, which is another database by the EBI. And here should be an option. There should be an option that allows you to download the sequence. So, sorry, I just need to find it. I'm not very familiar with this database. I think it's, no. Okay, let me look into this, but there should be, I'll get back to you on this. There should be a way to download the sequence directly from the Interpro entry. So let me just come back. So. Okay, I've just found it there. I realize it's a super family, so you need to select your the actual domain first. So what you do is you go here to the right-hand side, left-hand side, click on the feature you want, and then on the accession number. So you can see in this case, this is on a different bacterium. And then once you click on the accession ID, you can download the sequence here on the right. I hope that answered your question.
We can't hear you, ma. Hello. We can't hear you.
Can you hear me?
I hear you now.
Okay, brilliant. Okay, thank you for being back on time. That's amazing. I hope you can see my slide as well. All right. Sorry, I'm just looking for my window here. Okay, so welcome back. I hope you had a nice lunch break. I did. It was nice to stretch my legs a little bit. So we will move on with variation data in Ensembl. But before we move on, are there any questions at all? Okay, I don't see any questions in the chat box, and I also don't see any questions in the living document. So if you do have any questions, please feel free to put them down in the chat or in the living document, or you can raise your hand as well. Okay, so for this module, I will talk about how variants are classified, and then also the species for which variation data is available, and sources from where we retrieve the data from. We will explore the variation data in Ensembl during the demonstration. And in the end, you will again have some time for exercises to explore the Ensembl website yourself. So let's talk about variation types first. We can group variants according to their size or the number of nucleotides that are affected. So variations can come in a small scale where only one or a few nucleotides of a gene are affected. So in this slide, we have two sequences. We have the top sequence is the reference sequence, and the bottom sequence is our own data. So small scale variation types can include small insertions or deletions, which are also called dips or indels, or these can be single nucleotide polymorphisms. These are also called SNPs. And these are changes in the nucleotide. So for example, for this very first one, in our reference sequence, we have an A, but in our own sequence, we have a T. This would be a SNP, a single nucleotide polymorphism. For the second one, we have a C in our reference, but a deletion in ours. So this is an indel deletion. And then for the third one, we have a deletion or no nucleotide at this position and a G in our sequence. So this would be an insertion. Variations can also come in a larger scale, so in the chromosomal structure. And these are also called structural variations. So the types of structural variation are copy number variations or CNVs. So for example, when you have a genomic sequence with a lot of repetitive regions, and we also have large deletions, duplications, insertions, and translocations. So variation data is available in a number of different species across our Ensembl sites. In the Ensembl main site, we have variation data available for 36 different assemblies. In plants, we have 19 assemblies, variation data available for 19 assemblies. For metazoa, 19 as well. Protists, 2, and fungi, 8. We do not have variation data available for bacteria. So for the next few modules, I will actually be using fungi as an example for our demonstrations. So where does the variation data come from? In general, there are 4 steps in the Ensembl variation data process. So first, variation data is imported from a publicly available database. During the import, the data is being checked for quality. Then we compare the data that passed that quality control to other databases and link them with any associated studies or traits. And then finally, we analyze the data and process them to make them available in Ensembl. So let's look at each step in more detail. In the first step, we import variation data from a publicly available archive or source, and we search variation data from a number of different databases. So the logos that you see here are just a number of those databases where we import the data from. But in general, we take variants from the European Variation Archive or the EVA, or any species-specific projects and collaborations. In the second step, in the second step, we apply our quality control measures to the data that we imported. So here we map the variants to a reference assembly. We compare the reported variants with the reference assembly, and we exclude any suspect variants. So for example, any alleles that contain ambiguity codes or non-nucleotide characters. And you can find out more about the different quality control measures that we apply in this link at the very bottom. I've just noticed that my slide here is wrong. So this should be quality control and not variant import. In the third step, we link the variation data that passed the quality control to other databases. So what we do here is we add value to the data. For example, by linking a variant to any allele frequencies or to any traits and publications. In the final step, we apply our own analyses on the variation data that we imported and that passed the quality control. So here, we predict the variant consequence, the protein function, we calculate linkage disequilibrium, variant conservation across the different species. So how do we predict the variation consequences? So what we do, we take each variant and we plot it along the genome, and we predict the functional consequence of the variant depending on where in the genome the variant falls. So on the slide, we have a gene. We have these lines. So let's pretend this is a eukaryotic genome. We have these lines that represent intronic regions. We have these blocks that represent exons. And any empty boxes are how do you call them? Non-coding exons, sorry. And these yellow boxes are coding exons. So for example, for eukaryotes, if a variant were to fall outside the gene, so the 5' or 3' regions, the variant is unlikely to affect the gene itself, but it would likely affect the regulation, or so for example, the mRNA transcription and transcription activity. If the variant were to fall within the gene, but not within a coding sequence, so let's say an intronic sequence, so these lines here, the variant is also unlikely to affect the gene itself, but it might affect the mRNA stability, so splicing or translation efficiency. If a variant were to fall within the coding region of a gene, so these yellow boxes here, there are two effects that the variant could have on the gene. So the variant can have a synonymous or a non-synonymous effect. Sorry. If for synonymous variations, the protein sequence remains the same, so there is no change in the amino acid. But for non-synonymous or missense variations, there is a change in the amino acid and therefore a change in the protein sequence, and that means that it would pretty likely affect have a greater effect to the gene itself and the function of the protein. So to summarize, in Ensembl, we take each variant, we plot them along the genome, and depending on where the variant falls within a gene, we assign a consequence to that variant. Similar to gene functions, we also have special terms called sequence ontology or SO terms for variants. And this is just a standardized way of describing sequence-based features. Just like how we have a standardized way of describing a gene function. So I don't expect you to read this table at all, but what I want to show you here is that these sequence ontology or SO terms have very particular names and also color codings in Ensembl. Okay, so when we talk about non-synonymous or missense variants, we rank them by their pathogenicity. There are a number of different algorithms that rank these changes in the amino acid based on how conserved the protein is. It looks at the chemical change in the amino acid position and also the 3D structure of the protein. And for example, we calculate the SIFT score or the sorting intolerant from tolerance score. So this is a score that predicts the variant effect based on how well conserved the protein is. So SIFT scores are ranked, are on a scale from 0 to 1, with a score closer to 0, meaning a more deleterious effect of the variant, and a score closer to 1, a tolerated or benign effect. One important thing to remember is that these prediction scores are predictions and not facts. So a prediction will never be as good as experimental validation. So it's always good practice to validate your prediction experimentally. Okay, and finally, I also want to talk about reference alleles in variation data. So if you remember from this morning, I talked about how genome assemblies are made, that they are made of contigs, which make up scaffolds and then assembled into chromosomes. But what we also know is genome assemblies are a hybrid assembly of a genome. So each section comes from a particular individual. So when you take a section, so let's say we'll take a BL1 here, when you take a section of the genome assembly and look at a particular variant, so T in this case, it means that this T allele is found in this, in this particular individual. But it doesn't mean that the majority of the individuals have this T allele. So this individual might actually be might be carrying an allele that is that causes susceptibility in a disease. So when we talk about reference alleles, it doesn't always mean it's the major allele. So the major allele is the allele most frequently found in a population. Most of the time, if you talk about reference allele, it is the major allele, but sometimes it is not. So it's just something to keep in mind. Okay, so for this demonstration, we are going to look at the ADH4 gene in Saccharomyces cerevisiae. And what we're going to do is we're going to look at all of the variants within the gene. We're going to look at the variants within the region, so also the flanking regions of the gene. And then we're going to look at a particular specific variant in ADH4. So first up, I'm going to copy and paste what we were looking for into the chat box. And then we are going to move to our browser. So for this morning, we were looking at examples in bacteria, but because we don't have variation data available for bacteria, we're going to move on and look at variation data in fungi. So first up, I'm going to go to the Ensembl Fungi website. So for this, you just type in fungi.ensembl.org. And here you are on the website. So this is very similarly structured to all the other Ensembl sites. You can select your species of interest from this drop-down menu. So I'll do that. I'll search for Saccharomyces cerevisiae. So if you use your keyboard and just type in S, it will send you right down to the S section of the drop-down. Okay. Saccharomyces cerevisiae, and it's R6411 that we're looking for. Oh, can't find it here. Okay, so if you can't find it, like me, oh, I just noticed it's in the favorite genome section here, so the very first genome. But if you can't find it from the drop-down menu, you can make your life easier and click on the view full list of all species here on the left-hand side. And just type in your genome into the filter here. Oh, sorry. Into the filter in the top right-hand corner. There we go. So I've just typed in the assembly name, well, the first part of the assembly name, R64, and just making sure that this is indeed the genome we're looking for. So it's Saccharomyces cerevisiae, R6411. So I'll click the species name and enter my gene of interest into the search box. Sorry about this. Okay, so I'm entering ADH4 into the search box, and then I'll click go. And then similar to the bacteria, to searching for a gene in bacteria, we have this search results. We have the gene ID, we have a description, the species, and then also the exact coordinates. So we want to open the gene tab directly. So I'll open this, I'll click on this gene ID. And now we are on the gene tab in Saccharomyces cerevisiae. And so we've looked at gene information. Now we want to look at variation information. So here on the left-hand side, we have a section that is called genetic variation. And here you can find all of the variants within the ADH4 gene in either a table format or an image format, so plotted against a genome. We will look at the table format, so let's open the variant table link. And here on the right-hand side, you'll see a table of all of the variants of in our gene ADH4. So let's look at the table first. So we have different information about each of the variants. We have the variant ID, we have the location, we have the alleles. So the first one is always the reference allele, and the second one is the alternative allele. We have the class, so the type of variant. This is a single nucleotide polymorphism. We have the source, so this is the database where we took this information from. And we have the consequence types, so these are the sequence ontology terms, so terms of the variant. If it's protein coding, we also have information about the amino acids. And if it is a missense variant, so if it's a pathogenic variant, sorry, you will have a score here under the SIFT column. If you're not sure what each of the columns represent, you can always hover over the names and it gives you a brief description of what the data is showing you. You can filter this table. So you can use these filters up here. So there is a SIFT filter that you can apply, and there's also a consequence filter, or you can filter any of the other columns. So if you click this filter other columns, you select your column from this drop down, and then you enter how you want to filter it. Let's filter by consequence. So let's say you're only interested in missense variants in this gene. So for this, I would click consequences. By default, everything is turned on, so we're showing you all of the consequences. To select a single one, I'll turn all of them off and then select the one that I want. So missense variant in the second column here. And then you click apply to save your filter. And then you'll see our table is much smaller now, and we only have missense variants here. You can also sort the table according to a specific column. So you can click on the column name, and that sorts the table either ascending or descending, depending on what it is. And you can download this table. So there are two ways to download it. You can download it using this Excel icon in the top right-hand corner. So this will just download it as a table format. Or you can export data here on the left-hand side. So if you click export data on the left-hand side, what you're doing is you can export it in loads of other formats. For example, you can download it in the FASTA format, you can download it in any other annotation format, and you can also get it in the embed format. So if you want to download the table as it is, use this Excel icon. If you need your variants in a very specific format for any downstream analyses, use the export data format. Okay, so this is how you view all variants in a gene. Now let's look at how you can view variants in a region. So not just the gene itself, but also the neighboring regions. For this, we want to go to the to the location tab. So this is the tab right next to the gene tab. So I'm going to click that. And we land on our region in detail view. So we have, this looks a bit different compared to the bacteria location tab, because Saccharomyces cerevisiae has multiple chromosomes. So we are on chromosome 7 at the moment. This is the coordinates we're on. Our region of interest is highlighted in this red rectangle, not green. Next, we have this overview. So this is the 50 kb overview. Our gene of interest is highlighted in green, and the region is also highlighted in this red rectangle. And then scrolling down, we have a region in detail view. So this is just the region that is this red this rectangle here. And in this region, what you can do is you can add tracks. So you might have noticed that there is already a track in here that is called variant all sources. And if you're not sure what it is, what data it's showing you, you can click on the name and it gives you a brief description. So here we're already showing you all the variants within our ADH4 gene. I'm just going to make sure that there aren't any other variant tracks that I can add. So I will click on Add Remove Tracks here in the top left-hand corner. And then on the left, I'll select Variation. And we can see that there are only 2 tracks available for Saccharomyces variation data. So we have sequence variants, all sources, and SGRP variants. So because I already selected all sources, it's automatically going to show me the SGRP variants as well. So SGRP is a database from which we took the Saccharomyces variants from. So this means that I don't need to add any more tracks because we're already showing you all of the tracks available. So I can close the menu and view the variants in the region properly this time. So we have our variant track. Each of these blocks or lines represent a single variant. So this is one variant, this is one variant, and so on. We have 2 different colors, so 2 different variants available in our track. And if you look at the bottom, there is a legend for variants, and yellow are missense variants, and this green is a synonymous variant. So you can actually click on the individual variants and find out information that way. For example, I've just clicked on the synonymous variant. So we have the variant ID, we've got the class, so this is this single nucleotide polymorphism. We have the exact location, the alleles, so the reference allele is T, the alternative allele is C. And then we have the consequence as well, so the sequence ontology term. Okay, so that's how you view variants in a region. If you wanted, you can zoom out of the region in detail view to actually look at the, the neighboring, the flanking regions. So I've just zoomed out and you can view all the variants in the flanking regions as well. Or if you even wanted, you could look at variants of the flanking genes. So for example, MNT2 here. Okay, so we've looked at variants in a gene, we've looked at variants in a region. Now let's look at a specific variant. So for the specific variant, I'm going to go back to the species information page. So by clicking Saccharomyces cerevisiae here on the left-hand corner, and I'm going to enter the variant ID that I've given you. So it's S07-16069. And then once you're happy, you click Go. And we have our variant here. So I'm going to click on the variant ID. And this opens a new tab, actually. So this is now the variant tab. And in this tab, we have variant specific information. So here in the left-hand side, we have different variant displays or views. By default, we land on the Explore this variant view, and this just gives us summary information about the variant. So the consequence, we have the alleles, we have the minor allele frequency, the location, we have the variant in the VCF format, in VCF. So this is the variant consequence format. I need to double check that. I'm not sure what the C stands for, but it's a type of format where variation data comes in. We also have a list of HGVS names. So if you open that, you can find all the synonyms of this variant. And then, of course, we have the source of the variant. So under external links, the original source is SGRP. And then you can find more information about SGRP here in this link. So if you want, you can look at the variant in a genomic context. So here on the left-hand side, we have genes and regulation. So if you click on that, you can see where the variant falls within a gene. So sometimes a variant might fall within a regulatory feature, so you'd have information on that here. But in our case, our variant only falls within a coding region of the gene. So we have an entry here. This is the gene name. We have the allele, we have the consequence and the position information, and then also a SIFT score. You can always hover over the SIFT scores if you're not sure what the different numbers mean. So for this one, 0.1 means it's a tolerated effect or a benign effect. You can also look at genotype frequency. So here on the left-hand side, if there is population data available, which for this variant there is, you'll find information on that here. So the population in which they looked at this variant is the SGRP population. And in the population, what they found is the G allele is found in 97.4% of the population, so 39. This is the count, so 39 individuals. And oh no, wait, this this count does not look right. Ignore the count. Focus on the frequency here. I'm not sure why the count says 39 for all of them. I need to double check that. But if you wanted to look at the individuals of the population, so this is the population summary. But if you wanted to look at the individuals, you want to go to sample genotypes here in the left-hand side. So here we actually only have one genotype available to us. So we have this individual K11 and their this individual's genotype is A, and the population belongs to SGRP. Okay, so I think this is most everything I wanted to show you regarding variation data. So now we have time for exercises. If you go to the training page under variation, there are exercises for different species, just not for any bacteria. So please try them out so you can get familiar with finding variation data in Ensembl. I will give you 20 minutes for this, and then we'll come back and see how it goes, and then I'll give you more time, or we'll move on to the next module. And if there are any questions, you can put them in the chat box so I can see there's already one question in there, so I'll answer that. I didn't answer it during the presentation because I'm not actually sure about it, so I need to do some research on it. Or if you have any other questions, you can put them as well in the living document.
Thank you, Ma. We're going to come back after 20 minutes.
Yes, thank you.
Right.
Hello, we are ready. We are done with the exercise.
Okay, wonderful. Wow, that was very fast. Okay, so are there any questions before I move on?
No. Are there any questions from anyone? No. No.
Okay, wonderful. Thank you.
Thank you.
All right, so let's move on to the next module. So I am going to talk about the variant effect predictor, which we also call VEP. So what is VEP for? Or what is VEP in general? VEP is a tool that you can use to annotate and predict the functional consequences of your own variants. So let's say you just received your sequencing data and you've called variants from this data, and now you want to find out more information. So for example, the consequence of your variants. And this is what you can use VEP for. You can find out the effect of your SNPs, insertions, deletions, and any structural variants. Oh, sorry. So this is how you use VEP. First, you will need to put your data into one of these formats. So web can only work with these formats: variant coordinates, VCF, HGVS, variant IDs, or SPDY. I will show you examples of the different formats if you're not sure what they look like. And then once you enter your data into web, it is processed by the tool, and what it does is it searches Ensembl and any other external databases to give you information like the effect of the variant, any pathogenicity scores if it's a pathogenic variant, allele frequency data if there is any available, and also any publications. There are 3 different interfaces for web. There is the web interface. We have the command line interface, and you can also access web via the REST API. Today, we are going to look at how you can access or how you can use the web interface. So for the demonstration, we are going to look at 5 variants in Saccharomyces cerevisiae. These are the 5 variants, and we're going to use web to find out if the variants have already been annotated in Ensembl, what genes are affected by the variants, and also if any of the variants are missense variants. So I'm going to jump out of my presentation and go to the training page first, actually, because on the training page, we already put the variants into a format that web can recognize. So if you go under the web tab here in the training page and open the demo section, if you scroll down, there is a little section of the variants, and these are already converted to you in the VCF. So VCF is a variant format, variant calling format, and each column represents a different information about each of the variants. So one line is one variant. So we have 1, 2, 3, 4, 5 variants here. And the first column will tell you the chromosome. So in this case, all of our variants are on chromosome 7. We have the coordinate, the ID, the reference allele, and then the alternative allele. So what we'll do is we will copy this, and you can copy that as well on your screen. I'll also put it in the chat box in case you need it there. And what we're going to do is we're going to go and open up our Ensembl tab. So I've already got it open here. And you can access that from any page. So you don't have to go to the home page. What you need to do is you can click this tools icon at the very top of the page, so in the navigation bar. And here we have the different tools that you can use in Ensembl. The first one is the variant effect predictor, so we'll click that. And you can see here is where we enter our information. So first things first, we want to make sure that it's the correct species that we're looking at. So it's Saccharomyces cerevisiae, it's the assembly R6411, this is correct. But if you wanted to change your species, if you're looking at a different species, you want to select change species. And then you can scroll to the very top of this pop-up menu and enter your species into this search box. So if you're looking for Fusarium, for example, you can enter Fusarium. It will auto-complete for you. You can select it, and then it'll give you all of the Fusarium species we have available. But because our species is already selected, I'm not going to change anything, so I'll just close this menu. Okay, next we want to give our job a name. This is always good practice in case you look at your results again in future. It makes it easier for you to find. So give your job a descriptive name. In this case, I'm just going to call it servicia demo. Oh, it's very difficult. Demonstration. But you can call yours whichever you like. Okay, and then we have our input data section. So here you can enter your data in this box. We have examples for each of the variant formats that you can enter in web. So we have the Ensembl default format, we have VCF, variant identifiers, or HGVS notations. Especially for the ones that are formatted in columns, you might not necessarily know which column means what. In this case, you can click on this question mark in the next to variant effect predictor. And there is a link that says input format. And if you click that, you will find examples and then also a detailed description of each of the formats. So for example, the Ensembl default web format, it looks like this. And the first column represents the chromosome. Then we have the start, end position, allele, strand, and then also identifiers. I'm going to paste the link for the formats into the chat box as well. Okay, let me close this tab for now. If you have a file that you want to upload instead, so if you didn't want to copy paste, you can also upload a file. You can just click choose file and then select your file from the pop-up window. But the file should be no more than 50 MB in size. So if you have a file larger than 50 MB, you will need to save it online and provide the file URL instead. Or you can divide your variant file into multiple files. So make them smaller. Okay, so we have our variants already copied. So I will paste these into our input data text box. Next, we have any additional configurations we want to get. So each of these additional configurations are divided up into different categories. So we have identifiers, variance and frequency data, additional annotations, predictions, filtering, and advanced options. Under identifiers, you can see here next to it is a little description. So what you find here is what you can select here are any additional identifiers for genes, transcripts, and variants. So, for example, if you wanted VEP to tell you the ID of the protein within which the variant falls, you can select enable that here. You can also enable UniProt. How do you call it? UniProt entries here. So if you wanted VEP to give you a direct entry to UniProt, you can enable that here as well. Next, we have variance and frequency data. So the most important, I'd say, configuration in here is this very first one. So it's called find co-located known variants. And basically what this means is, do you want VEP to tell you if the variant already exists in Ensembl? And this is turned on by details. So what we're saying is, yes, we want VEP to tell us, does the variant exist in Ensembl? You can also turn this off if you like, but it's always nice to know if the variant has already been annotated or not. Next, you can also select some variant synonyms. So variant names can be ambiguous. So if you want it, you can turn this on. I will actually turn that on. And you can also tell Web to show you if any of the variants are flagged. Then we have some additional annotations. So you can, this is more relevant for eukaryote. Oh, this is more relevant if a variant had multiple transcripts. So we have different transcript flags for vertebrate species in Ensembl. And what they are is they just give you the confidence of the transcript annotation. Oh, sorry. No, I don't want to do this. Okay. Sorry, my computer just asked me if I wanted to update, but not right now. So you can enable these transcript flags in here. And then we have these prediction scores, so the pathogenicity scores that I mentioned earlier. By default, we show you the SIFT score, but we have a number of other scores available as well. And then if you're interested in any phenotype data, for example, in human, you can enable that here as well. And then finally, we have this filtering options section. So good practice is to not have any filters on before you look at the results, right? So because you want to see all of your results. If you apply a filter before you see your results, you might miss out on some interesting variants. So by default, this is turned off. You can turn on the filter if you like, but it's always good practice to look at your results first and then apply a filter. Once you're happy with your input, you can click Run here at the bottom of the page. And on this, so this page is refreshed every 10 seconds. You can see that here in this brown button. We have the name of our web analysis. So I've called it a Cerevisiae demonstration. And you can see it's running right now. So we're just waiting for web to process our variants. Here on the right-hand side, you have some options for your job. So you can save your web job directly into your account if you have registered, if you have an account on Ensembl. You can edit your query. So if you realize, oh, wait, I want to add these configurations to my query, you can click this and then you can just change your configurations that way. So you don't have to start all over again. Then you can also share, generate a URL to share this job with your colleagues. And finally, you can delete it if you don't need it anymore. Once the job is done, you'll see this green button, so that says done, and you can click on it to view the results. And these are our web results. So at the very top, we have some summary statistics. So here in this table, we have the number of variants processed. So we can see 5 variants have been processed, 0 have been filtered out. This is because we didn't apply any filters at the beginning. 1 variant is novel and 4 variants already exist. So 4 of these variants have already been annotated in Ensembl. The variants fall within 8 different genes. And we have these nice pie charts as a graphical, to show you graphically what consequences, what varying consequences we've got. So 50%, if we look at the coding consequences, 50% of our variants are missense variants, 25 synonymous, and 25 stop gained. If you scroll down, we have our table. So this is the more detailed results of our web query. We have a number of different columns. So the first column is our variant name. So this is the ID that we've given our variant, var1, 2, 3, 4, 5. You'll notice that there are multiple multiple entries per variant. And this is because a variant can fall on multiple genes. So if you focus on var1, here we have 1, 2, 3, 4, 5 entries for var1. And if you look at the symbol or the gene column, you'll notice that these are actually 1, 2, 3 different, 1, 2, 3, 4, 5 different genes. So this single variant falls on 5 different genes. And then we have the consequence of the variant. So depending on where the variant falls within a gene, you have a different consequence. So for example, for this very first gene, it's an upstream gene variant. If you hover over it, there is a description. Whereas this very last variant, this last var1 variant is actually a missense variant on this gene. We have the location in our second column. We have the allele. We have the biotype. So if the variant is protein coding or not. And if the variant is pathogenic, so if, for example, it's a missense variant, we also have a SIFT score available. So the table is quite wide, so you will need to scroll across the screen to see all of the columns. You can also hide columns. So at the top, you have this button that says Show Hide Columns. And some of these are already hidden, and this is just because there is no data available for them. So that's why they're hidden. But you can also show them if you like. So for example, impact, and then close it, and then you look for the impact. Impact column, and you'll see here that we have this impact here. And again, if you click on it, you can turn it off as well. You can filter the table according to, let's say, a consequence. So to do that, under filters, you select which column you want to filter. So let's say consequence, and then type in which consequence you want to filter. So let's say you just want to look at missense variants. You type in missense, you get a drop-down menu of the different autocompletes and click Add, and that will update your table. I'm just waiting for it to update here. Oh, there we go. So we've got 2 variants, var1 and 4. And these are the 2 genes, and they're both missense variants. And we have some scores here available as well. So if the variant already exists, we will have an entry in the existing variant column. So under here, and you can see that there is an ID available. So if you click on that ID, you'll see that there is an entry for this variant already available in Ensembl. Yes, and then if you want to delete your filter, you can also do that, of course, to get all of your results back just by clicking the X. And then if you want to download the table, you can do that in VCF, in the web format, or text format. So this is just a table format. Okay, so this is web. Now you'll have time to try it out for yourself. So in the training page again, we'll go up to the top of the page under web exercises. You'll find a number of different species again. So please try them out, and we'll come back in 20 minutes and see where we're at, and then I think take a little coffee break. Okay, hello everyone. Excuse me. Welcome back. I hope you had a nice short break. So before I move on to our next module, are there any questions? Okay, I don't see any questions in the, I don't see any open questions in the chat, and I don't see any questions in the living document either. So if you have any questions, please put them in the chat or the living document, or you can raise your hand to ask them anytime. Okay, so let's begin with our next module. So here I'll talk about comparative genomics data that is available in Ensembl. We're going to look at two main themes in this module. I'll talk about the applications of comparative genomics and the species available in Ensembl. I'll also introduce you to the comparative genomics data that we provide at the gene level, so gene trees and homology predictions. And then in the second part, I will talk about whole genome alignments and shared synteny. Okay, so let's look at the applications of comparative genomics first. So researchers in this field are mainly interested in examining the evolutionary relationship between species. And they're also trying to find and understand differences between genomes, derive a gene's function based on homology, and identify any regions that are conserved across different species. There are loads of different ongoing projects that generate sequencing data for researchers to do comparative analyses on. In Ensembl, we perform comparative genomic analyses by taxa. So this means that we compare all vertebrates against one another, all plants against one another, all invertebrate metazoa against one another, and so on. We don't perform these analyses on bacteria though, because there are just too many bacterial genomes available. And you can find comparative genomics data under the Gene tab, which we explored throughout the day today. We also perform pan-taxonomic comparative analysis, so comparative analysis across different taxa. For this, we take a number of representative species from each taxon. So some species from protists, some from plants, some from fungi, some from metazoa, vertebrates, and some from bacteria. And then we perform comparative analyses on all of those representative species. And you can find the pan-taxonomic compara data in the Gene tab as well, but under the pan-taxonomic compara category. We also offer comparative analysis data within species or within closely related species. So for example, we have mouse data, which includes all mouse strains and species available in Ensembl. And we also do this for rat. And we have similar comparative analysis data available for pig breeds and other agricultural animals. We can divide the comparative genomic data in Ensembl into two main types. So we have gene-based resources and sequence-based resources. Under the gene-based resources, you will find phylogenetic trees and protein families. So this would be under the Gene tab. And then for sequence-based resources, these would include whole genome alignments, for example, reconstruction of ancestral sequences. We calculate conservation scores and also syntenies. And these would be found under the location tab. So gene-based resources under Gene tab, sequence-based resources under location tab. A type of comparative analysis data that we produce are gene trees. So for this, what we do is we take a representative protein for each gene from each species. That's why we can also call them protein trees. We classify the representative proteins into families or some other kind of clustering method. Then we align them, we reconcile them against species trees to produce a gene tree. And then finally, from those gene trees, we can make homolog predictions and potentially infer the function of a gene in a different species. So this would be especially useful for species that are not quite as well studied as others, so non-model organisms. There are two main types of homologous relationships. So these are orthologs and paralogs. So here on the right, we have a simplified phylogenetic tree, or also called phylogeny. And, we have the phylogeny right here on the right. And then to explain these relationships, we named the different nodes, terminal nodes, in different names. Sorry, we've given the terminal nodes different names. So we've got H2, C2, H1, C1, and M here. So orthologs are genes that have emerged through a speciation event. And these are highlighted in blue nodes, blue internal nodes in the phylogeny. So for example, we have C1 and H1. These are orthologs because they have emerged from a speciation event at this node. Orthologs can also be described as one-to-one or one-to-many. So if you look at our phylogeny again, C1 and H1 are one-to-one orthologs because they're only orthologs to one another. So only the two of them. Then we also have one-to-many orthologs. So in this example, this would be M to C1, H1, C2, H2. So here's the speciation event. So M has one-to-many orthologs because M is an ortholog to C1, M is also an ortholog to H1, M is an ortholog to C2, and also H2. The second type of homologous relationships are paralogs. So these are genes that have emerged through a duplication event. And these are represented in this red internal node here. So for example, C1 and C2 and H1 and H2, these are paralogs. So C1 and C2 is a paralog, are paralogs, and then H1 and H2 are also paralogs. Orthologs often keep their function in the course of evolution, whereas paralogs may evolve any new functions. So to summarize, homology describes the relationship between genes and the events of how these genes have emerged. We display gene trees and gene gain or loss trees. So both are representations of how genes are related across different species. A gene gain or loss tree is a simplified or more collapsed version of the evolutionary history of a gene. And gene gain and loss trees are usually focused on whether a gene was gained or lost across different species. Okay, so we're going to look at a gene in Saccharomyces cerevisiae again. We're going to look at the TAS1 gene, and we're going to find its gene tree and also homologs to see if the gene has any homologs in other species. Okay, so for this, I'm going to go back to my browser. And I'm going to open my Ensembl tab here. And I'm going to start from the home page because this is a bit easier. I'll show you different ways of searching for things in Ensembl. Okay, so under favorite genomes, we have the Saccharomyces cerevisiae genome available. So I'm going to click that this link here, and I'll type in our gene of interest, which is TAZ1, TAZ1. We have our description, we have the gene name, gene ID, and then the species as well, and the location of the gene. And then we also have this link that says gene trees. So you can either click on this, or you can click on the gene ID, link. I want to click on the gene ID link because I want to show you the gene tree within fungal species first. So this gene tree is pan-taxonomic, so this is across different taxa, but I want to show you the gene tree within fungi only for now. So I clicked the gene name, gene ID, sorry. We have this gene tab open, and here on the left-hand side, we have two comparative genomics categories available. So fungal compara and the pan-taxonomic compara. Let's focus on fungal compara for now. So let's click on this gene tree link. So we're just waiting for it to load. Okay, took a bit of time, but it did load in the end. Oh, sorry. Oh no, I went back in my browser. So it has to load again. Okay, here we go. So we have our gene tree now. At the very top, we have some summary information. So we have the number of genes, which is 327, the number of speciation nodes. So these would be the event from which orthologs arise from, number of duplication nodes, so paralogs. We've also got the number of ambiguous nodes, gene split events, and you can also hide the annotations table. So the annotations table is just that table that I've just in there. If you scroll down, we have our phylogeny on the left-hand side. We have a graphical representation of the protein alignments. The first thing you might notice are these, how do you call them? The gray funnels. And these are just collapsed nodes. So we collapsed some of the nodes to make it a bit easier for you to look at, to make the phylogeny a bit easier to look at. So our gene of interest and species of interest is highlighted in red. So we have TAZ1 here, Saccharomyces cerevisiae. And if you click on one of those gray funnels, it opens this menu and it allows you to expand the subtree. Let's expand one of the smaller ones maybe, so it will load a little faster. So maybe this one here. So I'll expand this last triangle here, click on the funnel, and then click on expand this subtree. This will reload the page, and you'll see that this tree is now, well, the subtree has now expanded. We have the gene ID or gene name of our homologs here, and this is followed by the species name. next to it. If you scroll down, we have a legend as well. So, for example, our gene of interest, you can see here is highlighted in red. We've got any within species paralogs, so these are highlighted in blue. Here on the right-hand side, we also have different colored nodes. So the blue nodes are speciation nodes, the red nodes are duplication nodes. And then further to the right, we have different color codings for collapsed nodes as well. So the gray ones, these are the subtrees that are collapsed. So we just expanded one of them. We also have collapsed paralogs and then collapsed gene of interest if there are multiple entries for this gene. We also have a legend for the protein alignment. So anything that is empty means there is a gap. Then we've got this light green, which means 33 to 66% alignment. And then this dark green is 66 to 100% alignment. So you'll notice all of the collapsed nodes are the dark green ones, and all of the non-collapsed nodes are the light green ones. And that just means that for the collapsed nodes, there is a consensus alignment. So it's the alignment of this group of homologs. Whereas for the light green nodes, we have, these are one-to-one alignments. So these empty blocks are gaps. So there is no, for example, there is no alignment in this very first part of the protein. And then we've got these lines, these green lines, which means there is an alignment across the different homologs. You can export the image in different ways. So you can generate a URL and share it with your collaborators by clicking the share button at the very top. You can download the actual image that you are seeing. So if you click that, it will open a pop-up window, and you can select from different formats. So PDF, whichever format your report is in. So if it's a poster that you want to put this on, you can download it in a higher resolution. If it's just for a PDF, you can download it in PDF, and so on. And then you can also export the tree. So instead of exporting the image, you can also export the alignment. And there are different formats for these as well. If you scroll down, we have, similar to before, we have some previews of the different formats. So depending on the downstream analyses that you do, you might want, for example, a phylip format or a nexus format. So depending on what you're doing with this data, you might want to choose a very specific one. To select one, you can either click on the different images, or you can select this, click on this drop-down menu, select your format of interest, and then either download it directly or compress it first and then download it. So this would make it a bit easier. So someone's just asked, how do you convert phylogenetic trees to a pie chart? There are different ways to do this. There are programs where you just give it an alignment file and then it generates a tree for you, or you can do it yourself on R. So in RStudio, you can create a code that also allows you to change the different colors. So you can have your own color system and generate a tree that way, but it is much more complicated. I can give you some, some websites where you can just upload a file and then it will generate a tree for you if you like. Okay, so we don't want to download this alignment file. We'll close the menu for now. One last thing I want to show you here at the very bottom, we have some view options. So you have different options, different ways to view this tree, but you can also collapse nodes in bulk. So you don't have to click on the individual nodes at once. If you know you want to collapse nodes at the family order, for example, you can just select it from this drop-down menu here. If you select it, that will update the tree. Waiting for it to update there. Oh, let me restart this page. I just got an error that said it didn't load properly. While that's loading, I can answer the next question. So the next question is, can phylogenetic trees be constructed for a single isolate or sequence isolates? So if you have one isolate, you could get a number of closely related species and create phylogeny that way. It wouldn't be very useful to just display a single isolate on a phylogenetic tree, because you don't really have anything to compare it with. So it's always good to have a number of different isolates or closely related species in your tree. To do that, you would need to perform an alignment first. So there are different programs that you can do this with. All of the comparative genomics data in Ensembl, they're already pre-computed, so you cannot actually use your own data with the alignments here. But there are different tools available where you can perform alignments and create phylogenies that way. If you want to learn more about it, I can also look for some resources for that if you like. Okay, so it's finally reloaded, and you can see our tree is much, much bigger now because I've collapsed it at the family level. Okay, so this is how you view a gene tree. Now let's look at how you can view homologs of your gene. So I'll scroll back up to the very top of the page, and on the left-hand side, still under fungal compara, we're going to click orthologs. So if paralogs are available for the gene, this link would be an actual link and not grayed out. But because there are no paralogs for our TAS1, there's actually no information, so we can't click on this link. So I'm just waiting for the table to load. If you're ever not sure what the data that you're looking at is presenting, or if you want more information on how the data was compiled, you can always click on this question mark next to the name. Sorry about this, it's a bit slow. I think my Ensembl needs a bit of a break. Here we go. Okay, so the very first table is just a summary of the different species sets that you can choose from. So by default, we show you all of the species, but if you're interested in a particular one, you can click on these. You can select these here in this first column, in the second column. We have the number of and the different types of orthologs. So number of one-to-one orthologs in the entire set is 298. One-to-many is 14, many-to-many 0, and then without orthologs we have 58. You can scroll down to the bottom And you'll see this table, which has more detailed information about each of the orthologs. So we have the species name, we have the type. So let's just focus on this very first entry. It's a 1-to-1 ortholog. The gene name in the Obsidia rapens species is this very first link. You can compare the two regions, so you can compare A. rapens against our Saccharomyces region, and you can also view the sequence alignment. And then these last few columns give you some similarity statistics. And you can hover over the column names to get a description of what each of these means. Let's just quickly look at sequence alignment of two orthologs. So let's just select this very first one. I'm going to click on View Sequence Alignment, and then you can select from protein or cDNA alignment, so protein or coding alignment. I'm going to select the protein alignment. Okay, and now we have our sequence alignment for the protein. So this is in the clustal W format. This is in the clustal W format. We can see that here at the very top of the alignment. The first line is our reference, so Saccharomyces cerevisiae. And then the second one is the ortholog that we've looked for, so A. rapens. The different lines and stars, so the different symbols mean different things. If you're not too familiar with the clustal W format, so I would need to look up what colon and star and everything means, you can click on the question mark, which opens a new tab, and then you'll see here what the different symbols mean. So a star would be the amino acids are identical in the alignment. Colon, amino acids are different, but the function is conserved. And then a dot is amino acid is different and the function is semi-conserved. And then spaces, they're completely different. Oh. Okay, so you can download this alignment directly by clicking download homology, the download homology button. And similarly to before, you can select one of these formats and download it directly to your local machine. And the last thing I want to show you here is the pan-taxonomic comparer. So the comparative analyses, comparative genomics analyses we do across different taxa. So you can click, there are two different ways to look at these. You can look at the gene tree, you can look at the orthologs. So let's just quickly look at orthologs. This will give you a table similarly to the one to the fungal compara orthologs table that we just looked at, but it will give you orthologs across those representative species in each of the taxa. So I'm just waiting for it to load. Let me just hide this one for now because we don't need it at the moment. But if there is a species that you are interested in, for example, a human? No. Yes. If you wanted to find out if this gene, so TAS1, has an ortholog in another species, you can use this table, search for your species of interest, so for example, human, and see if there is an ortholog there. So for human, we do indeed have a 1-to-1 ortholog. This is what it's called in human. And this is the coordinates. And you can also view the sequence alignment for this. Okay, so let me go back to my presentation. We've looked at gene-based comparative analysis data. So we looked at gene trees and homologs. Now I want to show you how you can look at whole genome alignments. So whole genome alignments are especially useful if you want to find any highly conserved regions in the genome. So sequences that have evolved slowly and are therefore likely functional. They can include both coding and non-coding sequences, and whole genome alignments are also useful to support any problematic gene predictions or define syntenic regions, synteny, which I will talk about more towards the end. So in Ensembl, we have two types of whole genome alignments. We have pairwise and we have multiple genome alignments. For pairwise alignments, which you might have guessed, we perform them in pairs. So we take two genomes and we align the two genomes against one another. Across the different Ensembl sites, we have a number of these pre-computed alignments available. For human, we align the genome against all vertebrate species, and we also align model organisms against related species and agricultural mammals against one another. We also perform multiple alignments against predefined species sets. So we have sets of species where we perform those alignments on. Sorry. There you go. We have a number of species sets where we perform multiple genome alignments on. For the full list of these sets, just go to this link at the very bottom of the slide. But for example, we have one species set that is called the EPO analysis, and these include different species in fish, sauropsids, eutherian mammals, and primates. Okay, next I want to talk about synteny. So synteny is the conserved order of aligned homologs between species. So let's say we performed a pairwise alignment of human and chimp. And once aligned, what you will notice is that blocks of homologs actually keep their order. So if you focus on this very first block here, we've got the ALG11, UTP14C, NEC5, and NEC3 gene. And if you align this block against a chimp genome, you'll notice that the order of those genes is the exact same. So first ALG11, then UTP14C, then we've got NEC5 and NEC3. And this is what synteny is. So synteny means the order of homologous blocks is retained between species. And if you would like to learn more about our comparative genomics resources in general, there is a very useful paper here on this slide as well. Okay, so for this next demonstration, we're going to do a whole genome alignment in Fusarium solani. And this is the region that we're going to look at, and we're going to align this region to Fusarium verticilloides. Oh, I never know how to pronounce this one. And see if any of the regions align with the Solani genome. Okay, so let me just copy the coordinates and the species name into the chat, into the Zoom chat, and I will move to my browser, go back to my browser, and start from the Ensembl Fungi homepage. Okay, so we're interested in Fusarium solani. I cannot see it here on this under favorite genomes. So I can search for it from the species dropdown. So I'm going to type an F in my keyboard to find Fusarium. There you go. So Fusarium solani, select it, and then I will enter my coordinates into the search bar just underneath. So this follows the same sort of structure that we looked at earlier this morning. So it starts off with the chromosome number, then followed by a colon, start coordinate, minus, end coordinate. And once you're happy, you can click go. And you will land directly on the location tab. So here on the location tab, we'll scroll down to our we'll scroll down to our region in detail view. You can see that I'm actually zoomed in quite a lot. So what I'm going to do is reset the configurations here. Oh, that's not what I wanted. Okay, let me search for this, for the coordinates again. I don't think it searched it properly. Oh, that's weird. Oh, sorry about this. I've put the wrong coordinates into the slide. So the coordinates we actually want to look at is this. I'm typing it into the Zoom chat box right now. So just give me a second. Double checking. Okay, so these are the coordinates that we actually want to look at. So I'm gonna do the whole search again. Under Fusarium solani, I will type our coordinates and then click go. And here we go. Okay. So now we can see that we can actually see more than just one gene, one nucleotide here. So earlier I was just looking at one position, whereas now I'm looking at a region. Okay, so we have our region in detail at the very bottom. We have our genes in this region. So we are looking at the Fusarium solani genome at the moment. Now what we can do is we can add tracks to this. So we can add tracks to show the pairwise alignment of Fusarium solani to Fusarium verticilloides. You can find that under the Comparative Genomics category here on the left-hand side. And there's only one pairwise alignment available. So this is Fusarium verticilloides. You can click on it and select the style that you want it to be displayed in. I usually go for the normal style, but if you want it more compact, more succinct, you can select this second style here. So I click on normal, and once you're happy, you can select this check icon to save and close. So this will update our region in detail view. I'm just waiting for it to load, and it loaded quite quickly. So now you can see we have this new track that's called F verticilloides, and this is the alignment of our reference region to the verticilloides genome. Any aligned regions are highlighted in this kind of pink color. Anything that is empty means there is a gap, so there is no alignment here, for example. And you might notice that there are two rows. So there's this first row in verticilloides and the second row, and this just means there are two different chromosomes. So you can click on the different features. So I've just clicked on this very first block. And you can see more details about the alignment, but most importantly, you can find here, here you go. You can find here, it's very small. Hopefully you'll be able to see it in your screen, but these are the coordinates in Fusarium verticilloides, and you can see that it's on chromosome 2 here. Whereas if you click on the second row, you'll notice that this should be on a different chromosome. So this one is chromosome 4. Okay, so let's close this for now. Next, I want to show you how you can view an alignment as a text. So here on the left-hand side, still under the location tab, on the left-hand side, we have a link that says alignments text. And here is where you can see the sequence alignments of the two. So first you will need to select your species. So we only have one pairwise alignment, which is Fusarium verticilloides. Select that, click go. And you see a table of all of the aligned blocks between the two genomes. So we have two blocks, let's say, two regions that are aligned between the two species. So if you click on the very first block, you'll see, first of all, a preview of the alignment. If you want to view the whole alignment, you need to select Display Full Alignment, and you'll see the alignment file of Fusarium solani to Fusarium verticilloides in the region that we've just searched for. So in this case, it's this region here. You can also look at, you can also compare the regions in sort of a graphical representation. So if you select region comparison, you'll need to select your species of interest first. So again, Fusarium. And then what you have is those two, you have the region and detail view for those two species that we've selected. So here at the top, we have our Solani, and then at the bottom, we have the verticilloides one. And you can start making comparisons and see if there are any genes that are similar in the two species. If you scroll down, these aligned regions are actually highlighted in green. So you can see that the NASHA G gene aligns to this region in verticilloides. And then finally, you can look at a large scale comparison. So you can look at the synteny between the two. On the left-hand side, I will select synteny. So here we're on chromosome 2. I'm just going to make sure that I have selected the correct species. So I want to look at Fusarium verticilloides. Okay, and what we've got here is so the chromosome in the middle is our chromosome in Solani. And then here on the sides, we have the chromosomes in verticilloides. You can see it here at the very bottom as well. And any of these lines means that there is a syntenic region, that there is synteny between the two species. You can click on them to view the region more close up, if you like. Okay, so I think that's all I've wanted to show you for the comparative genomics part. So this was quite a long presentation and demonstration. I'll give you time for exercises now. I'll give you more time than before because there are quite a lot of them and it takes a bit of time to get used to finding the different gene trees and finding the right tabs. So I'll give you half an hour for this and then we'll come back, we'll see where we're at, and then we'll move on to the next module. Are there any questions before you start the exercises? So I think I've answered the ones in the chat box and I don't see any in the living document. So if you have questions, just let me know. I'll be here. Okay, it's 4 o'clock now. I hope you had enough time to do at least some of the exercises. Are there any questions before I move on? I don't see any in the living document and I don't see any open questions in the chat box either.
Thank you.
Okay. Oh, thank you.
We really followed up. There is no question.
Brilliant. Okay, so let's move on to our final module for today. So we're gonna talk about BioMart, which is a tool in Ensembl that allows you to extract loads of data at once without needing to know any programming language. So we just move on. Okay. All right. So what is BioMart? BioMart is a web-based tool to export Ensembl data without needing to know any have any programming skills. So if you want to extract big amounts of data, often what you will find is you will need some kind of programming or at least some kind of knowledge of command line to download that data. In Ensembl, you can use BioMart to get loads of data at once, and all you need is your browser. So you can build queries using a point and click method, so just with your mouse, and generate custom data tables and files that way. One thing that BioMart is not meant for is it's not meant for downloading whole genomes. So if you want to download, for example, the entire genome sequence of Fusarium solani, you will need to go to our FTP sites for that, where you can download the file directly to your computer. So why should you use BioMart if you can just get all of the information from the different Ensembl pages? So today we looked at all the different types of data you can find in Ensembl. We looked at gene information, we looked at variation information, we looked at comparative genomics data. So you can do that. You can go to the, sorry about that, there's an ambulance outside. Okay. So you can go to the different tabs that we visited today, just like going to specialty shops. So you can get your bread at a bakery, you can get your cheese at a cheesemonger, you can get your vegetables at a vegetable grocer. But to make it easier for you, you can just go to a single supermarket to get all of your groceries, right? So that's what BioMart is there for. It's sort of like a one-stop shop for all the different types of data you can find in Ensembl. On BioMart, you can query multiple genes and variants at once. You can convert IDs, you can extract coordinates, you can download multiple sequences in one go. So in other words, you can use BioMart for tasks that would be too time consuming by going through the Ensembl browser alone. You can find BioMart on Ensembl and Ensembl Genomes as well. So the structure of the browsers are the same. There is a shortcut to BioMart at the navigation bar at the very top of each page. BioMart is available for the vertebrate species in Ensembl, for plants, for invertebrate metazoa, protists, and fungi, but we don't have BioMart available for bacteria, unfortunately, because with over 3,000 genomes available for bacteria, it was just not feasible to do that. All right, so now that you know what BioMart is and what you can use it for, let's look at how you can actually use the tool. So you can use this 4-step principle to efficiently use BioMart or make a BioMart query. So for step 1, what you need to do is you need to define the database that you want to search in. So for the Ensembl main site, these will be genes, variation, regulatory features, or mouse strains. For Ensembl Genomes, I believe the only databases available are genes and variation. Next, you will need to define your species of interest. So this can be human, this can be Saccharomyces cerevisiae. So depending on whether your species is available, you can find that in the dropdown, which I will show you during the demonstration. Next, you will need to define your set of species or genes, your set of genes or variants by applying different filters. So these filters can be a region in the genome, a list of IDs, any particular function or trait. Basically, this is your input. So you can build very simple queries by applying a single filter only, or you can build more complex queries by applying multiple filters. For example, let's say you have a particular region in a genome that you're interested in, but you only want to extract genes that have a certain function in that region. So by applying multiple filters, you can narrow down your search, leaving you with a list of genes with common features. And this list is what we apply attributes to in the 3rd step. So in this step, you are defining the type of data that you want for the set that you've given in step 2. So you're selecting the attributes of your data output. For example, you can choose if your output should have things listed like IDs, any other features, variants. You can download sequences, and you can also extract orthologs from other species. And finally, you can view and download your results in a number of different formats. So these include HTML, tab separated, comma separated. Oh, sorry, this is supposed to say CSV, not CSC. And then also an Excel format. Sequences will only come in the FASTA format. One thing to watch out for if you do open your results on Excel using the XLS format, just be aware that sometimes Excel likes to automatically reformat some of the cells. So sometimes it likes to change gene names to, for example, dates. So it's just something to be aware of. And then if you would like to learn more about BioMart and Ensembl, we have these 2 papers that are very useful, that are very interesting reads. Okay, so let's move on with the demonstration, So we're going to get a list of Fusarium solani genes that are associated with very reduced virulence. So this is a pathogenic phenotype. But we only want genes that are not orthologous to Fusarium oxysporum. So we don't want any genes that has homologs to Fusarium. This list should include the coordinates, the functions, so Go terms, gene ontology terms. And then we also want the cDNA sequences of the genes. So let me just copy that into that information into the chat box, so I don't have to keep moving back and forth between my windows. So first, we want to move to our browser. So I've already opened the home page here, but you don't have to go back to the home page. You can just go to the BioMart link at the very top of any page. So this is Ensembl Fungi. Next, so step one, we want to select our data set. So this is BioMart. It's very simple, very, very minimalistic. We can choose our database from our drop-down menu. So here it's either genes or variations. We are interested in genes, so we will select genes. Next, we need to select our species. So we are looking for genes in Fusarium solani. So from the drop-down menu, we'll search for Fusarium solani. There you go. So you can type F in your keyboard, and that's just the second to the last in the Fusarium list. Okay, so once you've selected your data set, here on the left-hand side, you'll see that we have more options available now. So the second step is to apply our filters. So this is going to be our input. So I'll click on filters here on the left. And the very first thing I want to do is we want to find genes that are associated with reduced virulence. So reduced virulence is a phenotype, and you can find this under the pathogen phenotype tab on the right-hand side. So if you open this tab, it will give you some options. And the pathogenic phenotype is reduced virulence, so I will select that. You can you want to make sure that you've indeed selected it, so you have a tick mark here on the left. And you can also double check under filters if this has been added to your filters. So we can see pathogenic phenotype, reduced virulence is there. So next we want to find, we only want genes that are not orthologous to Fusarium oxysporum. So for this, we want to go to our to our multi-species comparisons. So that's the second to the last tab. And we want to look for orthologous Fusarium oxysporum genes from this list. So we have orthologous Fusarium oxysporum genes, but we don't want them. So we want to exclude them. So I'll just select exclude here on the far right, and just making sure it's been added to our filters. The check mark is there, and under filters, we can also find it here, orthologous Fusarium oxysporum genes excluded. Next, what I like to do is I like to count the number of genes. So there is a button at the very top top left-hand corner that says count. And this just counts the number of genes that your filter applies to. So we have, from this, we can see that we have 3 genes that have the phenotype reduced virulence and do not have an ortholog in Fusarium oxysporum. So there are three genes out of 16,163. Okay, so next our filters are done. We move on to the third step. We select our attributes. So again, on the left-hand side, under attributes, if you select that, you'll see that we have different categories available now. So we have the features, attributes, structures, homologs, and sequences. You can only ever select one category at a time. So because we want to get coordinates, function, and cDNA sequences, so we want features and sequences, we'll need to divide our query up into 2 separate queries. So let's get the coordinates and the functions first. So coordinates and functions are under features. The first thing you want to open is the genes tab, the gene tab. And here you can find some features, some Ensembl related features that you want to print in your results. So by default, we always print the gene and transcript ID, but you can also select your coordinates here. So we want to get our start and our end coordinates. So I've just clicked on them, and you can see here on the left-hand side that they've been added to our attributes. You can also add some other information like gene name or the source of the gene, but I'll leave it at this for now. So let's close this, this tab and open the next one. So the next one is the external tab, and here we can get information from external databases. So because Go terms, gene ontology terms, are from an external database, we'll need to select that from this tab, and we are going to select the accession ID and also the name. And again, you'll see, make sure that it's been added to your attributes. Okay, so I think we've got everything we wanted for features. So we want coordinates, we want function. So now we can get our results, our first set of results. So if you click results here on the top left-hand corner, you'll see on the right that it's populated your, sorry, how do you call it? That your results have been printed on the right-hand side. By default, we only show you the first 10 rows of the table. If you want to see the entire table, you can select from this drop-down menu all, and that will open a new tab. And it's not actually a very big table. So 12, it's only 12 rows. But it's always good practice to open all in a new tab just to make sure you didn't miss any interesting results. So, okay. From our attributes, we selected coordinates and functions. So what you might have noticed is each column represents one type of attribute. So we selected different attributes, and each of the attributes that we've added will be added in our results as a column. So we have our gene ID, transcript ID, start, end coordinate, the Go accession ID, and then also the Go name or description. What you might notice is we have multiple entries for each of the genes. So when we hit the count button, we actually only counted 3 genes. The reason why we have multiple entries for the genes is because Go terms can be, multiple Go terms can be assigned to a single gene. So you see here the very first gene ending in 37 has 1, 2, 3, 4, 5, 6, 7, 8. Wait, 1, 2, 3, 4, 5, 6, 7, 8 different Go terms associated with it. Then we've got the second gene, no Go term associated with it. And then the third gene, so there are 4 entries, and there are 4 different Go terms associated with it. So that's the reason why we have multiple entries for each gene. Okay, let's close this. If you want to download your table, you can do that as well. So there is a section at the very top of the BioMart tool that says export all results to. You'll need to select how you want to export it. So do you want to download the file? Do you want to download the compressed file? Or do you actually want to get it sent to your email? If you want it sent to your email, you just select that and then add your email address underneath. If you want to download it to your computer, select file or compressed file, make it faster. And then the format, so I would usually go for CSV, and then select Go. And that will open a new window for you to download the file to. Okay, so we've got our coordinates, we've got our function. Now we want our cDNA sequences. you can start from the very beginning. So there is a button that says new at the very left-hand corner at the top. But if you're lazy like me, I would just edit the query that I've already done. So you can click on attributes again on the left-hand side. And this time we want to select sequences, the sequences category at the top. So you'll see here on the left, the attributes have been reset. So now we can enter, select the sequence that we want. So under the sequence tab, you can select the different types of sequences you want to get. And we also have this little diagram that just reminds you what region of a gene you're about to download. This is more relevant for eukaryotic genomes. But we are interested in cDNA sequences, so we'll select cDNA here on the left-hand side, and you can see, on the right-hand side, and you can see that we've added that to our attributes. Okay, so once you've added that, you can hit results again, and you'll see that our sequences come in the FASTA format. So this starts off with our gene stable ID. So that's the first thing in our attributes. Then the transcript stable ID, that's the second thing. And then our actual sequence, the third thing in our attributes. By default, we show you the first 10 rows. So if you want to look at all of them, select all. That opens a new tab, and we will have our FASTA sequence in our new tab. So we have 1, 2, 3 sequences for each of our genes. Okay, and then finally, if you don't want to download the table to your machine, if it's, for example, a very big results table that you have, you can share the URL with your collaborators. So there is a link here at the top right-hand corner that says URL that generates it. And then what you want to do is copy the entire URL and send that on to your collaborators so that they don't have to do the BioMart query again. They can just enter that and then they'll get the results straight away. Okay, so that's BioMart. Now we have time for our last set of exercises, which is BioMart. So I will give you 20 minutes for this. We'll see where we're at, and then we'll come back and I will answer any questions if there are any. So I can see there are some in the chat. I'll answer them now. And then if there aren't any questions, we'll move on to some closing remarks where you can also get a certificate of attendance. All right, so I can see a question in the Zoom chat box. Just a second. I'm just getting the exercises on my screen. So the question is, how do I use BioMart to get the corresponding gene IDs? Is it a particular question you are referring to?
Yes.
Could you tell me which one it is? I can't find it right now. Oh, is it the convert IDs using BioMart, the second to the last question?
The second to the last question.
Okay, sorry about the feedback. Okay, so let me go through it in the browser so it's easier for you to see. So the very first thing we want to do is we want to go to the correct BioMart homepage. So in the question, it says, let me just open it here. So it's BioMart exercises, and then quite far down. So here it says BioMart is a very end, The following is a list of 29 human proteins. from the NCBI RefSeq database. So it's human, so we actually want to go to the Ensembl main site for this. So we go to ensembl.org and then use the BioMart link at the very top. And then you enter those codes, the RefSeq codes. Let me just show you really quickly. So you select genes because we have a list of genes. You select human because we're interested in human. So we're just waiting for it to load here on the left-hand side so we can select our filters. It's taking a bit of time. But if it has loaded for you already, oh, here we go. So we go to filters and then you go under the gene tab and you enter your RefSeq IDs in here. What you want to make sure is to select the type of ID that you're giving because Ensembl cannot recognize these automatically. So from the drop-down menu, you also want to select NCBI RefSeq. RefSeq, NCBI. So just double checking. NCBI gene accessions. There we go. And then you enter your genes here, and then you can get your results that way. Okay, it's 4:40 now. Would you like some more time for the exercises, or will I wrap up and finish for today?
Louis, I think you can go ahead and wrap up. There's no indication that anybody needs more time.
Wonderful. Yeah, so if you do want to go through the exercises again in your own time, they will be available to you forever, or at least until there is no internet anymore. Okay, so thanks everyone for staying until the end. The very first thing I want to ask you is for some feedback. So we have a feedback form on the training page. I'm also posting a link in the chat box. And I'll show you where it is in the training page. So if you go to the training page, scroll to the very top of the page, there is a survey and you just click that link. And basically what we just ask is for your honest feedback so we can improve our courses and make it even more helpful for future students. At the very bottom of the feedback form, there is a section that asks you if you want a certificate. So if you select yes, and then click next, it will be sent to your email directly. I'm going to give you a couple of minutes time to fill this out, and then we will finish off. All right, thank you for those who've already filled out the survey. I will finish off for today because it's already 10 minutes to 5. So let's just wrap up what we've learned today. I know it was a very long day, so hopefully after the workshop, you'll have time to process everything. But today we learned about genome browsers. Because genome data is really large and constantly changing, genome browsers are a nice way to show the information that have been published. So it integrates updated genomic information. It shows annotation information. So for example, any genes, variants, any motifs that are mapped onto the genome. And you can use genome browsers to get general information about any genomic feature. So genome browsers allow you to search, browse, retrieve, and analyze genomic data. Ensembl in particular maps genomic features to genome sequences, and we perform analyses on the data. Ensembl is what we call an added value resource. So what it does, it brings together information from a wide range of other databases to a single site. And other genome browsers you might have heard of are NCBI and also UCSC. We all might use the same reference genome, but the gene annotation methods may be different between the different genome browsers. So we looked at gene annotation data, we looked at variation data, we also looked at comparative genomics data. One thing we didn't cover today was regulation data, and this is only because it's only available to human and mouse. So the data is very limited for this, and in the pre-course survey, I saw that quite a lot of you were interested in non-vertebrate species. So that's why we didn't cover this. But if you are interested, let me know. I can send you some links on where you can learn more about regulation data and Ensembl. So we mainly looked at the Ensembl browser site. So we looked at the main site. We also looked at the Ensembl Genomes site. We played around with the BioMart tool a little bit. One thing I didn't show you was our FTP site. So this is where you would download whole genome information from. There is a link on the slide. So if you click on the slide, all you need to do is search for your species of interest in the table, and then you can get a download link directly for it. If you enjoyed the training, then please do recommend us to your colleagues. So apart from the Ensembl browser workshop, we also offer a more advanced REST API workshop. So this here we show you how you can access Ensembl data via the REST API. And it allows you to integrate it into your own pipeline. So if you have your own pipelines, the REST API might be really useful to you to integrate Ensembl data into your analyses. We also offer train the trainer courses. So here we train you how to train other people on Ensembl. If you are interested, you can email us at helpdesk@ensembl.org, or you can find more information in the training site. So training.ensembl.org/hosting. You can follow us on our social media. We've got Facebook, we've got LinkedIn and Twitter, and we also have a blog where we release any updates on Ensembl, and we also post any job openings on there as well. For any help and documentation. So these are a few links that will be quite useful to you. So if you're interested in bioinformatics in general, you can go to the EBI training page. So that's the very first link on my slide. They've got loads of free online courses that you can take in your own time. And this can be about anything. So they have metagenomics courses on there. They have variant calling courses on there. There's a course that's called Bioinformatics for the Terrified, which is an introductory course to bioinformatics and the different methods that you can use. So I'd say if you're interested in bioinformatics, definitely start with that one. That one's very, very useful. If you want any tutorials for Ensembl specific data, you can go to our Ensembl website, and under tutorials, you'll find loads of instructions on how you can access different types of data. And we also have recordings on our YouTube channel. And finally, if you have a very specific question that you don't know, that you can't find an answer to, you can always email us at helpdesk@ensembl.org. So we will always try to get back to you within a day to answer your question on there. So Ensembl is completely open source. So if you use Ensembl in your work, we ask you to cite us. This helps us to keep track on who is actually using our resources and what they use it for. So if you use the Ensembl main site, that would be this very first this very first reference. If you're using the Ensembl Genomes site, so the non-vertebrates one, that would be the very second reference on here. And then finally, I just want to say that the entire Ensembl team is actually made up of almost 150 people. So it's a great team effort to make this project possible. And with that, I'll finish up. I'll stop talking. Thank you very much for staying until the very end. I'll stay behind for a couple more minutes if you have any questions. And I will also monitor the living document for the next couple of days. So if you have any long questions, you can put it on there and I will answer them as well. Sorry. Okay, so thanks very much.
Thank you very much, Louise. I don't think we can thank you enough. We've been talking non-stop since the morning and we have been learning a lot, and we are really grateful for this opportunity to learn. And we hope to continue learning using the Ensembl browser and collaborating in the future, hopefully. Thank you once more.
You're very welcome. Yeah, if you ever want any more workshops in the future as well, just give us a shout and we will organize something for you.
Thank you.